Horseshoe crabs are marine arthropods of the family Limulidae and order Xiphosura or Xiphosurida, that live primarily in and around shallow ocean waters on soft sandy or muddy bottoms. They occasionally come onto shore to mate. They are commonly used as bait and in fertilizer. In recent years, a decline in the population has occurred as a consequence of coastal habitat destruction in Japan and over-harvesting along the east coast of North America. Tetrodotoxin may be present in the row of species inhabiting the waters of Thailand. Because of their origin 450 million years ago, horseshoe crabs are considered living fossils. Classification: horseshoe crabs resemble crustaceans, but belong to a separate subphylum, Cheliceraata, and are closely related to arachnids. The earliest horseshoe crab fossils are found in strata from the late Ordovician period, roughly 450 million years ago. The Limulidae are the only recent family of the order Xiphosura, and contain all four living species of horseshoe crabs. Carcinoscorpius rotundicada, the mangrove horseshoe crab, found in Southeast Asia. Limulus polyphemus, the Atlantic horseshoe crab, found along the American Atlantic coast and in the Gulf of Mexico. Tachypolis gigas, found in Southeast and East Asia. Tachypolis tridentatus, found in Southeast and East Asia. Anatomy and behavior: the entire body of the horseshoe crab is protected by a hard carapace. It has two compound lateral eyes, each composed of about 1,000 ommatidia, plus a pair of median eyes that are able to detect both visible light and ultraviolet light, a single endoparietal eye, and a pair of rudimentary lateral eyes on the top. The latter become functional just before the embryo hatches. Also, a pair of ventral eyes is located near the mouth, as well as a cluster of photoreceptors on the telson. Despite having a relatively poor eyesight, the animals have the largest rods and cones of any known animal, about 100 times the size of humans. The mouth is located in the center of the legs, whose bases are referred to as gnather bases and have the same function as jaws and help grinding up food. The horseshoe crab has 5 pairs of legs for walking, swimming, and moving food into the mouth, each with a claw at the tip. Except for the last pair, the long, straight, rigid tail can be used to flip the animal over if turned upside down. So a horseshoe crab with a broken tail is susceptible to desiccation or predation. Behind its legs, the horseshoe crab has book gills, which exchange respiratory gases, and are also occasionally used for swimming. As in other arthropods, a true endoskeleton is absent, but the body does have an endoskeletal structure made up of cartilaginous plates that support the book gills. They are more often found on the ocean floor searching for worms and mollusks, which are their main food. They may also feed on crustaceans and even small fish. Females are larger than males. C. Rotundicada is the size of a human hand, while L. polyphemus can be up to 60 centimeters long. The juveniles grow about 33% larger with every molt until reaching adult size. Breeding: during the breeding season, horseshoe crabs migrate to shallow coastal waters. A male selects a female and clings to her back. The female digs a hole in the sand and lays her eggs while the male fertilizes them. The female can lay between 60,000 and 120,000 eggs in batches of a few thousand at a time. The eggs take about 2 weeks to hatch. Shorebirds eat many of them before they hatch. The larvae molt 6 times during the first year. Raising horseshoe crabs in captivity has proven to be difficult. Some evidence indicates mating takes place only in the presence of the sand or mud in which the horseshoe crab's eggs were hatched. It is not known with certainty what is in the sand that the crab can sense, nor how they sense it. Blood: unlike vertebrates, horseshoe crabs do not have hemoglobin in their blood, but instead use hemocyanin to carry oxygen. Because of the copper present in hemocyanin, the blood is blue. Their blood contains amoebocytes, which play a role similar to white blood cells of vertebrates in defending the organism against pathogens. Amoebocytes from the blood of L. Polyphemus are used to make limulus amoebocyte lysate, which is used for the detection of bacterial endotoxins in medical applications. The blood of horseshoe crabs is harvested for this purpose. Harvesting horseshoe crab blood involves collecting and bleeding the animals, and then releasing them back into the sea. Most of the animals survive the process. Mortality is correlated with both the amount of blood extracted from an individual animal, and the stress experienced during handling and transportation. Estimates of mortality rates following blood harvesting vary from 3 to 15% to 10 to 30%. Fishery. Horseshoe crabs are used as bait to fish for eels and whelk. However, fishing with horseshoe crab is temporarily forbidden in New Jersey and restricted to only males in Delaware. A permanent moratorium is in effect in South Carolina. The eggs are eaten in parts of Southeast Asia and China. A low horseshoe crab population in the Delaware Bay is hypothesized to endanger the future of the red knot. Red knots, long-distance migratory shorebirds, feed on the protein-rich eggs during their stopovers on the beaches of New Jersey and Delaware.